this work, we describe the paternal and maternal ancestry of admixed urban populations from 3 Brazilian regions by studying the haplogroups of the non-recombining region of the Y chromosome and the mitochondrial DNA. Additionally, we analyzed a small sample of individuals from 3 Amerindian ethnic groups from the same regions, Guarani Mbia, Guarani Kaiowá, and Kaingang. Considering the whole Brazilian admixed sample, we observed a higher proportion of European Y chromosomes and Amerindian and African mitochondrial genomes. The more frequent European haplogroups were common Western European, concordant with the high contribution of Portuguese to the paternal gene pool of Brazil. Among the African Y haplotypes, we observed a higher frequency of haplogroups E, common in Western and Central Western Sub-Saharan Africa, corroborating historical records. The only Amerindian Y haplogroup detected in the admixed sample was QN3 that has been reported by other authors as the most common haplogroup in South Amerindians. Regarding the maternal haplogroups, we observed that the majority were Amerindian, mainly from clades B, but also C, A, and D. Clade B is very common in Amerindians from central western Brazil, from where our sample has more representatives, demonstrating the high level of admixture of our urban sample. Clades A and C are also present in central western tribes, but are more common in natives from the Tupi-Guarani branch, whose populations are spread by central western, southeastern, and southern regions in Brazil. The African haplogroup diversity was constituted by haplogroups L2, L3, L1, and L0 in decreasing order. Those haplogroups have a broad distribution in the sub-Saharan Africa, being particularly common in Western and Central Western Africa. The more common European haplogroup was H, which is also the most common in the Iberian Peninsula. Analyzing each region separately, we observed that the sex-related bias was maintained at this level. In Central Western and Southern regions, we observed a mainly European paternal ancestry, followed by African and Amerindian components in decreasing order. The Southeastern region, however, presented only European paternal ancestry. Despite a similar pattern was already described for this region, we suggest that the complete absence of African and Amerindian haplogroups may be due to the small size of our sample. On the other hand, maternal ancestry showed the opposite landscape. Central Western presented mainly Amerindian haplogroups, followed by African, whereas the Southeastern presented a majority of African haplogroups, followed by Amerindian. European and Asian haplogroups were detected at a lower frequency. In the southern region, the European component was the highest, as reported by others, but the African and Amerindian components together overpassed it. In this region was also detected a minor proportion of Asian ancestry. We also performed a principal component analysis based on the SNP genotypes from all admixed urban individuals in comparison with the 1000 Genome Project populations as references. We observed that the individuals grouped by shared haplogroup and confirmed the pattern exposed in the haplogroup analysis. Y chromosome Brazilian haplotypes concentrated near European samples. On the contrary, mitochondrial haplotypes showed a more uniform distribution, with a high proportion of haplotypes grouped near to Amerindian, African, and Peruvian samples. The analysis of an Amerindian pooled sample from the ethnic groups Guarani Kaiowá from Central Western region and Guarani Mbia and Kaingang from the Southern region also showed a pattern of sex-biased admixture. We observed 100% Amerindian haplogroups in the mitochondrial DNA and 80% Amerindian haplogroups in the Y chromosome, as reported by others. We found a European Y chromosome in a Guarani Kaiowá individual and a Sub-Saharan African haplogroup in a Guarani Mbia individual. The introduction of European haplotypes by the paternal lineage has been reported previously in the Guarani Kaiowá from central western Brazil, and here we can suggest the occurrence of admixture between Western European ancestry men, probably Portuguese, and Guarani Kaiowá women. On the other hand, the occurrence of admixture between Amerindians and African Brazilians have also been detected by others in Quilombo communities from the southeastern Brazil. Additionally, we could observe that the highest diversity of Amerindian maternal haplogroups was present in urban populations, suggesting that the admixed Brazilians are an important reservoir of native haplotypes, as reported in other studies. It is important, however, to highlight that our result could be limited by our small Amerindian sample size.